The perfect world. If I asked you to stop and think for a second what your perfect world would entail, would you imagine a world of perfect human beings? What if I told you that scientists are working towards creating that perfect human, and they're doing it all with CRISPR? CRISPR is a gene editing tool that is able to alter an organism's genetic makeup, aka their DNA. With a Cas9 protein and a donor DNA, scientists are able to effectively target DNA and modify the nucleotides present. Now, let's explore this. The Cas9 protein is an evolutionary tool that bacteria use to protect against viral infection. When viral DNA is inserted into a bacterial cell, the cell makes Cas9, which cuts it out, protecting the cell from accidentally producing any harmful viruses. Scientists manifest this tool by growing bacteria that produce more Cas9, and then isolate and attach a complementary guide RNA to help Cas9 recognize the sequence of interest for preceding modification. When inside the human cell, the Cas9 protein will begin scanning the DNA until it finds the target sequence to cut. But if there are copies of this sequence across the entire genome, how can we ensure that our Cas9 protein doesn't make multiple cuts and risk permanent damage? Well, it all lies with the PAM sequence. Cas9 will read until it finds 3 nucleotides in the form of any nucleotide, followed by 2 guanines that are adjacent to the sequence of interest. It will unwind the DNA and make sure that the strand with the PAM sequence is complementary to the guide RNA. Then it will make the double-stranded cut. While repairing the double-stranded DNA cut, the cell has 2 options: non-homologous end joining, which is sticking the 2 strands together, or homologous end joining, where the cell synthesizes a replacement double-stranded DNA through transcribing a customized donor DNA template previously injected into the cell. This new sequence will be inputted into the genomic DNA, containing the modifications that will change the overall genetic makeup of the individual. Now, why is this important? After modifying DNA, the new sequence read and expressed by transcription and translation enzymes will produce different molecules that change the individual's physiology. Thus, disease can be eliminated, individuals can become smarter and more athletic, crops can become bigger and more sustainable. The world can change to become perfect, but it's up to us to decide the limits of this power. Thank you.